Hello, Michelle Glass here. Welcome back to another episode for our chapter 18. Our topic here is still looking at hormones, and in this video, we're looking at the different mechanisms of hormone action. So I mentioned in the previous video that some of our hormones are going to bind to receptors on the cell membrane. So we can talk about those as extracellular receptors. Those are going to be receptors that are located on the membrane of your target cell. And we'll see often it's the amino acid and peptide-derived hormones that are binding to those extracellular receptors. An intracellular receptor is one that is found inside of the cell. And so we'll see that these can be located in the cytoplasm. We'll see these located in the nuclear envelope. We'll see them at the mitochondria, and we'll see them on the DNA molecule itself. And we'll see that it's most often those lipid-derived hormones that are going to bind to intracellular receptors inside the target cell, because remember, a lipid-derived hormone can travel directly through that phospholipid bilayer. Okay, so we've already mentioned that if we're talking about a G protein and second messenger system, that our hormone is going to be considered that first messenger. And the key thing to pay attention to here is that our hormone is not actually entering the cell itself, so this is how we can call it our first messenger. So we're looking here zoomed in at the cell membrane of our target cell, and notice that it has a hormone receptor that's fitting exactly perfectly with our hormone. And also notice inside of that cell membrane we have a G protein which is inactive. So as soon as that hormone is binding, that's going to activate our G protein, and it's essentially like the G protein is going, hey man, hormone's here. And then that is going to trigger our second messenger. So very often our second messenger is cyclic AMP. This is AMP here is standing for adenosine monophosphate. So you can see that an ATP molecule is used, broken down into an AMP. And then the cyclic part tells us that instead of it being a linear molecule, it's going to be a circular molecule. So in this drawing that I've given you here, we have our first messenger, which is our hormone binding to the receptor, an extracellular receptor. It's on the membrane. That's going to activate our G protein, which in turn is going to trigger our second messenger. Our second messenger here, cyclic AMP, is going to then go on down the line and either maybe open up ion channels or maybe activate enzymes, both of which would cause the cell to be stimulated. If you open up ion channels, you're going to get positive charge moving in, you're going to be depolarizing your cell. If you activate those enzymes, then you're changing the type of metabolism that's occurring in that cell. Sometimes our hormones, instead of activating our cells, inhibit our cells. So in that case, you would still have a first messenger binding to the extracellular receptor, activating our G protein. This time, the G protein is talking to the cyclic AMP, converting it just to linear AMP so that that second messenger is basically inhibited. It's not fitting in and opening any channels, is not activating any enzymes, so that's going to be slowing down the activity or stopping the activities of that cell. So these discussions here, the G protein and second messenger system, are those that involve those extracellular receptors, those receptors in the cell membrane of our target cells. And we often see our first messenger hormones here are going to be the amino acid derived or the peptide derived hormones. Our second messengers that we saw, what cyclic AMP was the example that I gave you, but calcium ions can also be an important second messenger. Those are the two most popular. They're not the only in the body, but they are the two that you're most likely to see described for you in your textbook. You also see, remember, that those lipid-derived hormones are able to travel directly into the cell, and so they're going to bind to an inside cellular receptor. And these receptors can be located, as I mentioned, in the cytoplasm or in the nucleus. So it could be the nuclear envelope or it could be on the DNA molecule. And these are going to be involved in triggering gene activation. So remember, if you activate your genes, you're changing the enzymes that you have available in the cell. When you change the available enzymes, you're also changing the types of chemical reactions that that cell can do. And then we can also see intracellular receptors as part of the mitochondria. And in that case, then you can see these hormones are going to increase the ATP production of the cell. And that's it for now.